Hello, class. Long time no see. Well, first of all, congratulations on all of you for completing your instrumentation sack. Hopefully, Briscoe didn't screw you over too much with that one. But I know it must have been tough without my videos on MMR and some of the other analytical techniques, but nevertheless, hopefully you all did fine. From what I've been hearing recently, it seems like most people seem a bit unconfident following the first couple of lessons on this new area of study on food chemistry. So I think now would be a good opportunity for everyone to consolidate their knowledge and address any gaps in their understanding, as it is quite a big area of study. But you'd be glad to know that the content, in my opinion, is relatively straightforward. But anyways, how this is going to work is I'm going to be breaking down the whole of food chemistry into 4 videos, each one talking about a different macromolecule. So today we're going to be talking about proteins. But first, let's have a read of the study design dot point. So it says proteins, formation of dipeptides and polypeptides as condensation polymers of two amino acids, primary, including peptide links, secondary, tertiary, and quaternary structure and bonding, distinction between essential and non-essential amino acids as dietary components. So what we have here is the general structure of the building blocks of proteins, also known as an amino acid. More specifically, an alpha amino acid or 2 amino acid. Well, we call it an alpha amino acid because this carbon right here is called the alpha carbon, and 2 amino acid because the side chain, which is this thing right here, but more on this later, is attached to the second carbon according to the UPAC nomenclature. Well, you've already probably heard the name amino acid before, but you probably don't know why it's named amino acid. Well, it's named amino acid because it contains an amine group which has a prefix amino right there, and the acid just simply comes from the carboxyl group being a carboxylic acid. And remember how I was talking about those side chains before? Well, this group largely determines the specific properties of amino acids, such as its acidity, basicity, and polarity. Well, now let's look at a couple of examples. So, as you can see here, we have 4 different amino acids, and each one of them displaying a different property. So, if you look up here, we have arginine first up, and from the abundance of the amine groups here on its side chain, it gives it the ability to accept protons, hence classifying it as a basic amino acid. And as you can see down here, this is going to be the structure of arginine in an acidic solution. Now remember how an acidic solution has an abundance of H plus ions. So and you can accept an extra proton right here and here to form a net charge of +2. And notice here how the carboxyl group next to the alpha carbon does not change. This is because in an acidic solution, the carboxyl group can neither donate or accept an H plus ion. And down here next we have aspartic acid, which by its name suggests, and the carboxyl group here on its side chain is an acidic amino acid, meaning that it can operate as a proton donor. Now this is the structure of aspartic acid at its intermediate pH, which is called a zwitterion. And this is just going to be the formal definition of a zwitterion, which is a molecule or ion having separate positively and negatively charged groups. And you need to know that the intermediate pH is basically the pH at which the amino acid forms its zwitterions. That is going to be a net charge of 0. As you can see, a negative here and a positive here is going to cancel to form 0, meaning it does not have an overall charge, obviously. Hence why in the case of aspartic acid, the carboxyl side chain is not touched, as you can see here, to allow the charges to cancel. Now, this goes for all amino acids as well. The formation of a zwitterion only affects the carboxyl and amine functional groups directly next to the alpha carbon, and the side chain is unaffected. Now, you want to be careful about something here. The intermediate pH, right, does not mean a pH of 7, as some places would say, although it can be for some amino acids, but that is not its formal definition. But in fact, each amino acid has its own specific intermediate pH. But the good thing is you'd be glad to know that you won't need to memorize them for each specific amino acid. Okay, now here we have serine, which is classified as a polar amino acid, as you can see from its hydroxyl group here on its side chain. And in a basic solution, the structure of serine is going to look something like this. And remember, basic solutions has a high abundance of hydroxide ions, which can accept a proton donated from its carboxyl group right here. And lastly, we're going to have alanine, which is a nonpolar amino acid, as the methyl group attached can only form dispersion forces, thereby making it nonpolar. So from the examples above, here, you probably realize that an amino acid can act either as an acid or a base given different conditions. So remember from units 1 and 2, we can refer to them as amphoteric substances. But that's pretty much all of the properties of an amino acid that you're going to need to know. So now let's get on to the formation of di and polypeptides. So here we have a simple diagram explaining the process of the formation of peptide bonds. Now, given that you've already studied reaction pathways, I'm expecting you to be pretty confident in how this works, but let's do a quick recap anyways. So using this diagram here, the OH group from the carboxyl reacts with the H from the amine to form water here. And the remaining products join together via condensation polymerization to form, in this case, a dipeptide, as you can see here, which is just going to be two amino acids joined together by an amide bond highlighted in red here. Now, also be careful to only use the term peptide bond when describing the bond between two amino acids. Peptide bond cannot be used as a direct substitute for amide bonds, so you're likely going to lose a mark if you call something a peptide bond given a different context. So I personally always refer to them as amide bonds as to avoid silly errors because you can never be marked down for that. Now, a polypeptide, as you can probably guess, is just going to be many amino acids joined together by amide bond, which will eventually fold upon itself to form a functional protein. Now, there's also going to be something called the N-1 rule, which is mainly used in calculations. Basically, what this is the number of water molecules expelled as a result of the condensation reaction between N number of amino acids. So, for example, we have two amino acids here, and the N-1 rule basically suggests that since we have two amino acids, 2 minus 1 equals 1 water molecule produced. So this is going to be especially useful when you have loads of amino acids joined together and you ask how many water molecules that it's going to form. So now let's get on to the concept of essential and non-essential amino acids. So all you're going to need to want to know is that non-essential amino acids are amino acids that cannot be synthesized by the body, and that essential amino acids are amino acids that can be synthesized by the body. Pretty straightforward, right? And obviously here are several examples of each, but again, you won't have to memorize these. And now you also should know that in order to have a balanced diet, that is a diet that covers all our nutritional requirements in a day, for example, we should eat foods that contain those essential amino acids that our bodies cannot naturally produce. As often a deficiency from these type of amino acids can lead to specific health-related diseases. And if you're vegetarian, then these amino acids can be attained from a variety of vegetables to cover a balanced diet, as one type of vegetable usually lacks some amino acids, as shown here, some examples. But the easiest way of attaining those essential amino acids is going to be just eating through animal products, as they complete the complete set of essential amino acids that you require. So now let's talk about the structure of proteins. Now, all my biology people will probably already know the importance of this, but just in case you're one of the smart ones not to choose bio, the structure of the proteins is important as it is directly related to its function because it determines whether the protein can interact with other molecules. But let's have a read of the study design dot point for this. So it says enzymes as protein catalysts, active site modeling of process by which enzymes control specific biochemical reactions, lock and key and induce fit models, consequences of variation in enzyme substrate interaction, lock and key mechanism due to the behavior of a particular optical isomer, explanation of effects of changes in pH, formation of zwitterions and denaturation, increased temperature, denaturation, and decreased temperature, reduction in activity on enzyme activity with reference to structure and bonding, action of enzymes in narrow pH ranges, and use of reaction rates to measure enzyme activities. So let's see how this might work. So, again, for the bio kids, you can feel free to skip this bit. But, anyways, what an enzyme is, it's that it's a protein slash biological catalyst, obviously used to speed up reactions. And in comparison to some inorganic catalysts you might have studied previously, enzymes are used specifically to one specific reaction, as a substrate must be geometrically complement to that of the substrate. Now, don't worry if you know what that mean. Don't know what that means. I'll explain it to you later. And since it's only specific to one type of reaction, this will typically lead to a greater rate of reaction compared to a generic catalyst. Now, let's take a look at this diagram, for example. Enzymes have a region in its structure called the active site, which is just going to be this region right here, that is used to bind to the substrate. Now, what a substrate is, it's basically the molecule that binds to the active site to get the reaction catalyzed. Now, the general gist of how this works can be demonstrated by the diagram here. So, a substrate forms weak temporary interactions with the active site of the enzyme here, and it forms what is called a enzyme substrate complex, and the enzyme lowers the activation energy of the reaction by weakening the bonds in the substrate, hence allowing the reaction to proceed at a quicker rate. But we'll talk more specifically about this in a bit. And also keep in mind that enzymes can distinguish the difference between enantiomers, meaning that usually only one enantiomer is biologically active. Because remember, enantiomers are mirror images of one another but cannot be superimposed upon one another. Therefore, if one isomer is geometrically complement, it does not mean the other isomer will also be geometrically complement to the same enzyme. But remember that enzymes can catalyze reversible reactions. And if you're interested in finding out more about this whole enantiomer business, you can look up the story of thalidomide. So now let's talk about the two types of models on how an enzyme might work. So first here we have the lock and key model, which by its name suggests that the enzyme functions sort of like a lock and the key is pretty much is substrate, meaning that the substrate's shape must be exactly complementary to that of the enzyme for it to function properly and catalyze the reaction. And then here we have the induced fit model in which the enzyme has a more flexible structure, allowing it to fit the shape of the substrate to catalyze its reaction, even though the shape might not be exactly complementary to that of the enzymes. And now will also be a good time to talk about the idea of coenzymes, which the study design dot point says the principles of the action of coenzymes often derive from vitamins as organic molecules that bind to the active site of enzymes during catalysis, thereby changing the surface shape and hence the binding properties of the active site to enable function as intermediate carriers of electrons and or groups of atoms. No specific cases required. So the study design dot point has pretty much encapsulated all you need to know about coenzymes. But how coenzymes might help catalyze a reaction with the addition of an enzyme is, for example, if you have a substrate that is of slightly different shape to an lock and key enzyme, a coenzyme can be added to change the shape temporarily of the active site of the enzyme to so that the substrate can fit in and the reaction can be catalyzed. And this can also work for the induced fit model as well. So we've talked about the two models of enzyme function, and now let's get into the factors that will affect enzyme function. So first we have temperature. You want to know that each enzyme has a different set of optimal conditions that will allow it to work at its best. So that is the quickest rate of reaction. For temperature, if the temperature is increasing before the optimal temperature is reached, as you can see by this bit of the curve here, then so will the rate of reaction of the enzyme. However, when the temperature continues to increase past its optimal condition, this bit of the curve here, the protein slash enzyme will eventually get denatured, which is essentially the process where proteins lose their secondary, tertiary, and quaternary structures due to pH and temperature change. But more specifically on this later. Whereas on the other end of the spectrum, if you decrease its temperature, the rate of reaction will decrease as according to collision theory, but it will not change or damage the structure of the enzyme in any ways. Hence, its effects on rate of reaction is only temporary, as the enzyme will restore its normal function when you increase the temperature again. And another factor that will affect enzyme function would be pH. Now, every enzyme has an optimal pH, just like temperature, where it works best at. But this time, unlike temperature, the enzyme will get denatured if pH is at either ends of the spectrum. So it can either be too high above the optimal, or too below the optimal. All right, so let's get on to the most important part of today's lesson. That is the different hierarchical structures of proteins. So first, we're going to have the primary structure of the proteins, which is going to be simply defined as the sequence of amino acids linked by peptide bonds in a linear polypeptide chain. So as demonstrated by a simple diagram here. Now, the secondary structure is defined as the primary structure of polypeptide chains folded into different shapes as a result of hydrogen bonding within the polypeptide chain. Now, you want to know that there are two types of secondary structures, alpha helices and beta pleated sheets. So what an alpha helices is, it's maintained by hydrogen bonds formed between the oxygen from carbon to oxygen double bonds within the hydrogens of N to H bonds, four amino acids down, forming a helix shape. And a beta pleated sheet is formed by linking two or more adjacent strands of the polypeptide by hydrogen bonds between oxygen and nitrogen oxygen groups of other peptide bonds. Now onto the tertiary structure, which is the structure that provides the protein with its unique 3D geometry and which ultimately gives its function. So it's basically going to be the overall shape of a polypeptide chain due to an interaction between R groups, such as hydrogen bonds, ionic interactions, disulfide bridges, and dispersion forces. So one disulfide bridge here is basically going to be a covalent bond that forms between two adjacent sulfur atoms. And finally, here we're going to have the quaternary structure, which is going to be the combination and interaction between two or more three-dimensional polypeptide chains. It's important to note that not all proteins have quaternary structures. Polypeptide chains in a quaternary structure is just going to be called a subunit. And a common example of this is going to be hemoglobin. Now, for the final part of the video, let's talk about the metabolism of proteins. So, first of all, what is metabolism? Well, metabolism is just going to be the breakdown and conversion of food into energy used by the body. So how this works is basically the food you eat contains proteins, and the pepsin enzyme in your stomach catalyzes the hydrolysis reaction of proteins into soluble amino acids. Now, given that you studied fuels, I'm relatively confident that you know what a hydrolysis reaction is, so I'm not going to be doing a recap here. But the amino acids have to be soluble so that they can be absorbed into the gastrointestinal tract and transported throughout the bodies to cell. And once they're there, they can be reassembled into proteins by condensation polymerization, which is the reverse of hydrolysis, to form proteins when it's needed by the bodies. But that's basically just going to be a short summary of proteins in food chemistry. Now, I know it's been a long time since I posted, but that's because of SACs and stuff. But please do leave in the comments any new videos that you want me to upload, or you can just find me at school to ask me any questions if you need.